Thank you very much. And welcome, everybody. Thank you for taking some time out of your busy conference schedule to come and hear the presentation. What I'll present today is our Streamlined CDx program, Streamlined Companion Diagnostics program. But I also want to reflect on two other observations. One is by Bill Gates, who observed or made the observation that people always overestimate what they can get done in a year, but they vastly underestimate what you can get done in a decade. And we've been around for 2 decades, so I'd like to show you what we've accomplished. And also, I'd like to say that this also integrates with what the investment community would say. Even though we don't have any venture capital investment in our company, we're private, and we built as an organic company. They say build what matters, and what I'm hopeful that you'll come away with today and what I can convince you of today is exactly that we built what matters over these two and a half decades almost. So the agenda is a brief company introduction, and then I'll introduce the Streamline partnership model that facilitates and accelerates drug approvals. And then I'll give you a little background and a little more information about some of our, I think, best-in-class assays that are out there already in kitted form with bioinformatics to over 700 customers in 160 countries. We've been profitable for more than 2 decades. So as far as our structure, we have new headquarters in San Diego. We took over an old Qualcomm building and gutted the interior and built it out to our specifications. And then we also have offices in Provence and in Munich, and we have an office in Shanghai that's been around for about 2 years that is already supporting clinical trials in China. And then we also have our Lab PMM clinical trials network of ISO 15189 accredited or compliant labs around the world that are have been involved for years in enrollment and testing of patient samples. So many of the assays, when a sample hits our labs, about 96% of those results are reported out within 48 hours. So this is just a more geographical representation of our laboratories and our offices around the world. In San Diego, in Martins Reed, and in La Ciotat, Provence, and then in Tokyo and in Shanghai. And we really do have state-of-the-art facilities. These, I've had comments back that these look a little bit like stock images, but in fact, those are actual photos of our new laboratory in San Diego, and people have remarked that no labs are that clean, but those are the real thing. So we're very proud of our new facilities. And we will announce shortly additional facilities both in La Jolla area and also in China. So the Streamlined CDx model, let me get to that. So as you know, in the companion diagnostics arena, being able to identify with a targeted therapy the appropriate patients really improves the clinical efficacy of the drug, and it can really accelerate approval because you might be able to end some of these trials early if you can demonstrate to the regulatory authorities that your treatment is working. And I'll also point out in this model that not only are the companion diagnostics better for identifying patients, but just moving from LDTs that are often used for enrollment to a companion diagnostic that is developed under design controls with good analytical backing and then GMP manufacturing at one location and then distributed has been proven to increase the clinical efficacy of drug during trial. We actually saw a 13% improvement with Novartis's drug, midostaurin, going from LDTs to bridging study with our CDx. So that was very helpful for our partner. So the challenges of CDx development are that generally you're moving with the pharma partner through various different phases. You start in feasibility, you have phase 1, phase 2, phase 3, and then you get into the regulatory submissions, both on the CDER side in the US and the CDRH side in the US, or the PMDA, MHLW in Japan, or alternative regulatory agencies. But then you also have to commercialize. So oftentimes in the traditional companion diagnostic arrangement, a pharma partner has at least 2 other partners, has the IVD partner, and then you also have the CRO testing for enrollment and clinical services. And we would argue that actually that's sometimes 3 additional partners, so 4 partners, because you have to also then commercialize the test at the end of the approvals of the device and the drug. So our Streamlined CDx model, we actually perform all these activities and have demonstrated that over the last few years, and I'll give you some examples of that. So the pharma's team, from the very inception of the first feasibility and phase 1 studies through commercialization, you're dealing with different team members within that pharma partner's company. But in Invivoscribe, our teams are very much vertically integrated. So you're doing development, you're doing manufacturing, you're doing the testing services for enrollment, and then regulatory and commercialization. Those are all activities that we're very familiar with 2.5 decades of work. So here's another schematic of how that works. The pharma, you have the non-clinical development, you go into clinical development, and you go into commercialization. Those are discrete silos generally within a pharma company, especially a big pharma company. But in our team, in Invivoscribe and LabPMM, our teams are very much integrated. So the commercialization team is already talking with the R&D groups and the clinical labs very early on in the process. So with our Streamlined CDx structure, you can see that on the product development side, we have over 2 decades of experience developing assays. All of the assays and the bioinformatics that are associated with them are all developed under full design controls. So ISO 13459 and also FDA design controls. And then we have comprehensive LimpTrack clonality products with bioinformatics software. These are already kitted and already sent out, and we have over 100 customers on the NGS space and have full freedom to operate on the Lumina platforms and the Thermo Fisher platforms. And then we have multiple NGS gene panels as well. As the agencies and pharma are also now asking for minimal residual disease assays that complement the diagnostic assays so you can track the patients over time. And hopefully we're seeing more and more that the regulatory agencies are more amenable to using MRD as a potential surrogate endpoint, which would really collapse some of these drug trials. On the manufacturing side, we're CDRH, FDA registered. You can see the other capabilities here on the diagnostic and companion diagnostic manufacturing site. We also have over 50 CE-IVD products in Europe or outside North America. And we've recently gotten over 60 of our assays, including our LimpTrack clonality and MRD assays with bioinformatics, approved in Australia through the ARTG. And we also have a whole selection of DNA RNA controls, MRD controls, proficiency panels, ASRs, GPRs. On the clinical testing side, we have a global reach. So we have we're supporting over 140 enrollment sites worldwide. We have a dozen years of clinical reference lab experience. Internationally standardized testing, which was the basis of the founding of our company, to have standardized testing reagents available worldwide with everything under design control. So they're all ready to be going through the regulatory authority. And on the quality, regulatory, and commercialization, we really excel in that we're experienced worldwide. We've done, you'll see here in a second, successful submissions to the FDA, the PMDA, MHLW in Japan, South Korea, Saudi Arabia. We're working with regulatory agencies worldwide. And in China, you'll see that we're moving forward very well as well. In China-based, right now in China-based clinical capabilities, we're working with the NIFDC on getting our assays approved for then use at the AAA hospitals. We're in AAA hospitals there. And we're moving to expand that reach in China. And we're working with partners for enrollment in clinical trials. And we've been there for 2 years. And as far as the China-specific regulatory capabilities, you can read them here. So we have an extensive knowledge. We've actually been working with the Chinese authorities because, as you might be aware, the landscape is changing a little bit. They're learning, and we're working with them, so we're right lock in step with them. So that's going very well. We're US-based, but we also have Chinese office and Chinese employees, and they're managing and auditing the AAA hospitals and working with the NIFDC on the assays as well. So, and we hold medical device licenses through the Shanghai office. We're also the market authorization holder for our assays in Japan. So this is an example of, living up to what I'm suggesting is our strategy here, which is in using our Lucastrat FLT3 companion diagnostic assay. We've gotten 3 drugs approved with 3 different partners in the last 3 years. So we worked with Novartis on midostaurin for FLT3 positive AML. We recently got approval both in Japan and the US with Astellas with their drug for FLT3 positive AML. And then with Daiichi Sankyo, we've completed submission both in Japan and in the US for their drug, quizartinib, for FLT3 positive AML. So this is just a little bit of our testing services. We have a whole range of gene panels that are quite remarkable, and we, of course, all our bioinformatics is also designed in-house. We don't offer any tests, any bioinformatics. Anything that we offer as a test or a support, something that supports a test, we developed in-house from the ground up under design control, so they're ready to go through the regulatory agencies. And we have a whole series of assays that go from initial diagnosis, big panels of 194 genes, or even larger, down to our lymphotract assays that can get down to 10 to the minus 6 as far as limited detection, just really dependent on how much DNA, how many cell equivalents of DNA, and how much sequencing depth that you're interested in doing. And here's some recent papers. This one's actually in press from Memorial Sloan Kettering Cancer Center. They've been using our assays now for a couple of years. And actually, if you look at the red, let's see if I have a pointer here. You'll see in red at the lower left there that initial assessment clonality was detected in greater than 97% of all malignancy types. So for all leukemias, lymphomas, they were able to identify a clonotype in over 97% of all cases. And then they can track them, if you see the blue bars in the upper right, from everything from plasma cell neoplasms like multiple myeloma through things like mantle cell and B-cell ALL. You're picking up with one assay, which is our IGH Framework 1 assay, you're getting almost 100%, and you can track those. So this is a very good paper, and I would recommend you read it. This is a paper out of Cornell, and it shows, if you look at the boxes in red, that our assays, again the same assays, picked up MRD earlier than multi-parameter flow. You can see the black arrows are for flow, and the red arrows are for our MRD tests. And here's a paper we did with Johns Hopkins, Mark Levis, who's a leader in AML. And this simply shows that our MRD assay for FLT3 ITD clearly stratifies patients. He uses this on clinical decision-making every day for his patients. And finally, this is a paper out of Oxford using our same assay, IGH Framework 1, to show a clear stratification of CLL patients on the basis of somatic hypermutation rate. And again, our assays not only identify clonality, they also call out and give you an automatic read on the somatic hypermutation rate. So I thank you for your attention, and happy to take questions after the session. Thank you.
And thank you, Dr. Miller. Good talk.